So very good day to everybody present here. This is information technology in bioinformatics and genetics. In today's session, we'll discuss about cloning, gene cloning strategies. In gene cloning strategies, we discuss about genomic DNA libraries, about the PCR, it's an alternative to genomic DNA cloning, and then we discuss about cDNA, complementary DNA sequences, okay, and libraries. So this is especially for you, my dear students and young researchers, and you can reach me at dr.christoananth@gmail.com. So before beginning the session, let me thank God for giving me this golden opportunity to deliver this useful session to share my knowledge among my fellow national, international participants, students, and young researchers. So in today's session, we'll have a short introduction to genome sequences. Then we discuss about complementary DNA sequences, about the PCR test, and then we'll discuss about generalized overview of cloning strategies. Then we discuss about requirement of the number of clones, about the PCR and alternative to genomic DNA cloning, and about the proofreading enzyme. Then we discuss about some genome amplification also. Okay, and then we discuss about splicing, then about cDNA, complementary DNA library, then finally we end this class with the lambda insertion vectors. So I've already posted the open-ended project work. Please complete them. As I told you, you have to complete both the independent works and the open-ended project work. So at the end of the class, I'll be giving you a short video to discuss the knowledge in our topic. Right. So whenever you're talking about genomic sequences, whenever you're talking about genes, so generally we will have OTS, that is nothing but off-the-shelf cloned genes. So here we will be analyzing the complementary DNA sequences for different organisms, maybe like monkey or maybe human beings or maybe whatever living organisms that has evaluated. We will be analyzing with the help of different characteristics of mammals. Okay. So here we will be categorizing a laboratory model for the entire species. So maybe it will have like domestic mammals or maybe the mammals that you analyze it for the phylogenetic trees. Okay. So this is the reason why you tend to use single gene cloning over multiple gene cloning. Okay. So these genomes or maybe the gene sequences, you are going to map it together. So when you map it, you will analyze the characteristics, you will understand why this single gene cloning is very important. Okay, so maybe let us take in the case of a polar bear. Okay, let us take a gene from a polar bear. So maybe it lives in the snow. Okay, right. So maybe only thing, whenever you are going to analyze the sequences, you can, from the polar bear, you can analyze it only in the single gene level. Okay, so genomic sequences or maybe genome sequences, it is going to reveal only some part of the information of that given gene. Okay, right. So these are the cDNA sequences, complementary DNA sequences. We will be analyzing from the mRNA sequences. So here we analyze the cell types, the stages of different development, and of course, for example, if I maybe use a pin and prick him, okay, maybe he will say he'll shout. So this is the stimulus that I'm going to give. So based on the the single gene sequences, okay, accordingly that stimulus is being got. Okay, so this is the experimentally stimulated external stimuli. Okay, so in this cDNA sequences, we will be analyzing some useful information, some part of the information, and we will be analyzing the splices, some important parts. For example, in the human body, you have cells. Each cell will be analyzing, you will be getting the information about the DNA sequences of that particular human being. So from the splices only, you get the useful information about the cDNA sequences. So from different tissues, from different development stages, you get different characteristics. So in the genes, you will have expression profiles or maybe biochemical functions also that will be giving more information about the genes. Okay, so this is how you tend to get information from the single gene analysis. Okay, and then you will have structural annotation phase. So that also comes in the gene function and formation. So altogether, it's not like one process and you get all the information. It's a collective process. You tend to get each of the information. Here we will have clone library. So we'll have genomic DNA library. So you'll have genomic DNA. So that's what is given in the diagram. And then you're going to isolate the DNA from the cells. Then we will have plasmid vectors also. Okay. So this is a cloning strategy which is based on the cell. So it's a cell based cloning strategy. So what you try to do, you divide the source DNA into different fragments, different pieces. Okay. And what you do, you try to clone everything. Okay. So I hope you know the meaning of cloning. Okay. Maybe you are trying to um, make a duplicate out of this one. Okay. For example, even in the movies also we have seen this one. Maybe in order to escape a particular problem, they do the cloning and they change the face and they try to do this one. Okay. They try to behave in a different way. So this is how it goes. Okay. So a collection of clones, generally you can go with the starting population called as the library. Maybe you can categorize it with the original clone and the different clones with which are separated. So this is PCR test. You should understand it's the polymerase chain reaction. Okay, polymerase chain reaction. So maybe it's going to get it from the source DNA. And what you have to do, you have to conduct this test. Okay, so in order to sequence directly from this source DNA, and you're going to split it, and the split parts you're going to combine it as a clone. Okay, so this is how it goes. This is a polymerase chain reaction. Okay, so every strategy has its own advantages and disadvantages. Of course, you have a screening also to carry out for this entire DNA population. So in the PCR approach, generally the first step is actually screening. So the fragments are closely analyzed, and of course you will go with the sequencing from the source DNA and cloning it to the individual fragments. So these are the genomic DNA libraries. So genomic library, whenever you say library, it's a collection of genes. Okay, so in the genomic library, you tend to collect several genes. So for example, here we will have ecori. Okay, I'm showing this one, ecori. So it's Escherichia coli RY13-1. So that's what you call to be ecor 1 or maybe ecori. Okay, you can pronounce it in any way. Okay, so this is a test generally where you insert the fragments of maybe a suitable vector. Maybe it will have lambda vector, and then for example, okay, somebody has stolen my bottle. Okay, here. Okay, somebody has stolen my bottle here, and I don't know who has stolen my bottle. Okay, now what I will do, I will ask this professor, one professor who came now. Okay, I'll ask this particular professor, come, try to find out. Okay, or maybe I will ask one of your friend from this class, go, just keep quiet and just look for who has took my bottle. Okay, so what I'm trying to do, I'm inserting one particular fragment. Okay, and see, he'll be very quiet, he'll be looking around, and he will identify who has took the bottle. Okay, and he will be taking that particular clone out of it. So this is what you do: insert the fragments into a suitable phage lambda vector, and then attempt to isolate the desired clone. So who has took the bottle, taking the desired clone. So this is how it goes. So maybe for this one, Escherichia coli RY13-1 gives maybe an average of some fragments of core 4 kb size, and generally this size of the human genome is in the region of 2.8 cross 106 kb, and generally we will have like 7 cross 105 independent recombinants. Generally it should be screened together in order to get that desired sequence. Okay, so this is how it goes with the genomic DNA libraries. So this is a generalized overview of cloning strategy. So we'll have like DNA fragments. So generally we'll have restriction endonuclease digestion. Then we will have mechanical shearing, duplex cDNA sequences, direct chemical synthesis, and then we will have PCR, polymerase chain reaction. Then joining to vector is the next step. We will go with the homopolymer tailing. Then we'll go for litigation cohesive termini. We go with cohesion. Then we go with the blunt end litigation. Then linker molecules and TA cloning as well. Okay. We'll have tail agent. Okay, tail agent cloning. Then you'll have introduction to host cell, transfection into with the recombinant phase DNA, then transformation with the recombinant plasmid DNA, and you go with the packaging. Then finally we go with this one, selection and screening, hybridization, PCR, immunochemical, protein to protein interactions, protein to ligand interactions, functional complementation, and finally the gain of the function. So these entire process go with the um, you know taking that particular desired DNA sequences and getting into the cloning procedure. So far in the previous slides, we discussed only about the single gene sequences. So here we will discuss about whole gene clusters also. Maybe if you take in the case of a gene, it can be cut. Okay, maybe internally it can be cut maybe one time or maybe two time or maybe more times also by this E-core 1 or maybe E-core I technique. Okay. So but if you try to do that, okay, so you will not get the single fragment, okay, because the gene is actually very large. Okay, so maybe you can go with the whole gene clusters also. So what you have to do, this extensive region you are going to make it up as a gene, or maybe you can make it up as a single gene or maybe as a whole gene clusters also. So for this, as I told you in the previous slide, see this one, 4 kb size. The fragments are having 4 kb size. So fragments averaging about 4 kb size. In this case, for the whole gene cluster, it's actually very short. Okay, so what you have to do is the gene may be contained on the E-core 1 fragment that is larger than the vector. So maybe in this case, the appropriate gene cannot be cloned. So only if it is small. For example, if you are a small boy, I can teach you what to do. If you are a big boy, I cannot teach you what to do. Okay, right. So this is how it goes. The appropriate gene would not be cloned at all. And generally, you can go with the cloning the random DNA fragments of a large size. For example, maybe your friend, for example, you are Yamon. Okay, you are Yamon. Let us take in the case. But your friend is Yakshi. So I will tell his this particular say, please take care of him. He's young on. Try to give advice for him. So if I say like this, then he will give advice to you. You will also be actually. You get it? You get it? So this is how it goes. So I cannot make you understand, but I will tell through him. Please make him understand, and he will tell to you, and you will be good. Okay, so this is how it goes with the cloning the random DNA fragments of the large size. And since the DNA is randomly fragmented, there will be no systematic exclusion of any sequence. Okay. So in this case, you can go with the cloning of the all sequences. So we go with the requirement of the number of clones. We will have clones which overlap with one another, and of course, because of the large size of the clone DNA fragment, few clones are actually required for a complete library. Okay, so for the complete library, if you try to do in this way, only few clones are required. So if you take in the case of a human genome, its size is actually 2.8 cross 106 kb. Okay, so the average cloned fragment size is actually 20 kb, and n would be the number of clusters would be 1.4 cross 105. Okay, so generally it's greater than n because the sample variation will lead to inclusion of these sequences, and definitely there will be less exclusion of sequences from this genome. Okay, so as I you can see in the diagram, so this is the genome to be stored in the library. Okay, genome to be stored in the library is cut up with the restriction enzyme. Maybe it can join with the recombinant phage DNA or plasmid, and of course there is plasmid library also where we will have the phage clone for each of the vectors. So human genomic DNA library, we will be categorizing the value of n. So n is equal to ln, ln. Okay, it is not log, but it is ln. Okay, ln 1 minus p divided by ln 1 minus n. Okay, so maybe if you have to achieve 95% probability, so maybe you will have this one, n is equal to ln 1 minus that p you are going to substitute as 0.95. So 1 minus 0.95 divided by ln 1 minus 1 by that n is actually I told you 1.4 into 10 power 5. So this one you are going to have 10 power 5. Okay, so you are going to have this one, and you will be getting the value of n is equal to 4.2 into 10 power 5. Okay, so generally if you have to achieve 99% probability, so in this case what will happen, ln 1 minus 0.99 divided by ln 1 minus 1 divided by 1.4 into 10 power 5. So in this case your n would be equal to 6.5 into 10 power 5. In 95% it is 4.2 into 10 power 5, but 99% it is 6.5 into 10 power 5. So this is the apt value for the human genomic DNA library. So naturally we can conclude that this PCR polymerase chain reaction, it's a robust technique for amplifying the specific DNA sequences from complex sources, and generally you are going to amplify short products. So that is the greatest advantage that you have. And of course maybe the product size that you have is around 5 kb. So the typical size is around maybe 1 kb to 2 kb. So that is the typical size. And generally with the case if you generally analyze the PCR enzymes, it's having poor processivity, and generally it will have a name called tac polymerase. Okay, so this is how it goes. And this is the the diagram with the case of long PCR. So when long PCR is going to use mixture of enzymes, it's going to amplify more DNA templates as well. So if you go with the extreme conditions, maybe if extreme conditions can cause serious conditions like damages to the bases, and of course you will have some changes, some alterations in the DNA strands. So as I told you, polymerase processivity is actually poor when you take in the case of TAC polymerase. So modifications you can do with in order to improve this polymerase processivity, and definitely you can increase the pH value, like partial value of hydrogen. Okay, partial pH is partial value of hydrogen. Okay, so that has to be increased. So if you increase the pH value, so generally you can protect it from the damage of the bases. Okay, so maybe if you use this condition for maybe 2 DNA polymerases, so maybe you will use one for the proofreading enzyme, and maybe you can generally go with the proofreading enzyme to remove the mismatched bases. Or if you try to remove the students who are lazy from this class and take it out, definitely the class would be good, right? So in this case we try to remove the mismatched bases from this one, what you call it to be the TAC polymerase. Okay, so TAC polymerase would generally will have this proofreading activity. It will be correcting them, and definitely you will try to amplify DNA fragments up to a size of 22 kb. Okay, so this is how it goes with the proofreading enzyme. And fragment libraries can be prepared from the material that is unsuitable for the conventional library cloning. So here PCR is the the apt strategy for the gene isolation. So you can isolate the gene from the PCR test, and of course the genomic libraries will be having like single cells from the fixed tissue that is coming up. So for this one we can use random primers. So random primers means it's a you can use this particular strategy for selecting size of the sequences. Also for suitable PCR products also we can go with the random primers. So here we will have linkers which are ligated to the ends, and generally we will have linkers that are associated with the primers. So this primer along with the linker, it is going to go with the size selection and suitable PCR product in order to go with the gene isolation. And we finally we go the whole genome amplification. So Here in the previous cases we analyze only the shorter sequences, so the whole genome amplification is actually necessary for amplifying by the PCR test. So for this one, shorter fragments will have very less usefulness, but if you go on a large scale, definitely it will be very much useful. And for this one we use MDA technique, multiple displacement amplification technique, so that we will be having a highly processive DNA polymerase sequence. So that definitely we can get more useful information by this MDA, multiple displacement amplification method. Okay, so here in the previous case we had product length only up to 22 kb, but here we will have the product length between 20 kb and 200 kb. So that much size it can process. And definitely you can go with the construction of the genomic library, and you can employ this PCR techniques ranging from 103 to 106. Earlier case it was very less, but now up to 103, sorry, not 103, 10 power 3 to 10 power 6 it will have. Okay, so in the previous case it was 10 power 5, but here it is 10 power 3 to 10 power 6. So this we will be having like cDNA libraries. Of course, that is a topic of concern. We will have clone eukaryotic cDNAs, which will be having like non-coding sequences, and corresponding to that particular DNA, we will be getting the DNA, gene isolation. So here we will have something called a bacteria that you call it to be introns. Okay, so generally we will be in the high eukaryotes, you'll be having this particular bacteria. So generally, in order to analyze this one, we'll establish a collinear relationship between the gene and the encoded polypeptide. So maybe in the untranslated region of around 5 or maybe 3 inches, you will get this particular bacteria. So they are copied into the by the RNA polymerase, and definitely you can go with several processing events in order to analyze and isolate the genes. Then we'll have a technique called splicing. So in order to remove this bacteria, remove this introns, we will have a technique called splicing. So maybe some genes, okay, some mammals, in mammals also, some genes will be having this larger bacteria called introns. So it will be present most. Okay. So maybe for example, if you take in the case of human dystrophin gene, generally it will have 79 introns. So maybe it will have more or less 99% of the sequences. So maybe the gene would be approximately 2.5 MB in length. It's not KB. We earlier talked about 22 KB, 25 KB, but now because of the introns, it will be having 2.5 MB in length. Yet, definitely the corresponding cDNA is over 11 KB size. So that's the advantage of the cDNA technique, complementary DNA technique. Okay, so you'll use this cloning for determining the size of the cDNA clone, and definitely it is lesser than the genomic clone. With the case of eukaryotic cDNA clones, generally you will have this, getting the primary expression of the polypeptide product, and we will be analyzing the boundaries, which is concerned with the intron as well as exon. Okay, so intron is nothing but the bacteria, exon is the outside portion that is available. So if you compare this intron exon boundary with the cDNA sequences, you can easily isolate the genes. Okay, so maybe in order to prepare the cDNA, you'll use the purified mRNA sequences, and generally you can screen for the particular and suitable clones. So in the cDNA library, you will be having the population of the RNA sequences, and you will get the the contents of the cDNA libraries. You will isolate the DNA sequences, get the gene isolation done, and for this technique only we use the splicing technique in order to remove the introns, and you can isolate the gene sequences as well. From this we will have ovalbumin. Okay, ovalbumin. That's actually from the mRNAs. We will have something like a egg white protein. Okay, from for example. Okay, similarly even it can be similar to the the white egg that you put it as a Okay, in the pan you put it, you'll have a white content, right? So that's like maybe you can assume like this one. It's a egg white protein. Okay, protein that is present in the mRNA sequences. So this mRNA will be enriched with this white protein. Okay, so that's what you call to be the ovalbumin. Okay, I'm showing this in the diagram. Okay, so here to obtain such abundant cDNAs, okay, where we will have mRNA with good white protein. So generally we will clone them directly into the M13 vector, what you call to be M13 MP8. So that's actually the vector that you will be used to get this abundant mRNA with the white protein. Okay. Then finally we go with the lambda insertion vectors. So based on the amino acid sequences for maybe 19 abundant sequences. So we were able to identify 13 out of 19 proteins. So that is only because of the going with insertion of the lambda insertion vectors. So just that's what I'm telling you. So you insert the vectors and you find out, you isolate the genes associated. Okay. So for the isolation of the cDNA clones, we will have the moderate as well as low abundance classes also. So we will not use it only for the low classes. We will be using for the low as well as moderate classes also. And generally from this you can construct the entire cDNA library. So that is the major advantage of the lambda insertion vectors. So we'll go with the phage lambda vectors. You can obtain the large number of cDNA clones. Okay. So lambda insertion vectors are very much suited for the cDNA cloning, and of course you can use these vectors for cloning procedures. So we'll also have xenopus oocytes also. That's also we'll have like analyzing different clones, 105 clones in mRNA sequences. So we were able to identify 15 molecules per cell, and maybe for few specific cells also we can do that. So you can go with the whenever you are going to prepare this mRNA, we can enrich this entire process, and you can go with the size fraction process, and maybe you can test the fractions for the pressure of this desired molecule. Maybe you can insert this mRNA fractions into xenopus oocytes. You're going to split it up, and you're going to categorize the molecules that are associated. Fine. So this is the open-ended project work. Please complete them. Please do them at your best available time. It should be your own project, and I will be looking into each one of them.